Good afternoon. I'm Dr. Penny Dacks, AFTD Senior Director of Scientific Initiatives. Since 2015, AFTD grant programs and research partnerships have driven more than $18 million in funding to FTD research. Investing to advance the science of FTD is critical to our goals as an organization. AFTD works in collaboration with people in pharma, academic institutions, government agencies, and other nonprofits to accelerate research, deepen the understanding of FTD, develop better diagnostic tools, and pursue effective disease-modifying treatments. The next few sessions of our conference will highlight collaborations advancing FTD research. Sessions will focus on progress in understanding FTD, potential treatments on the horizon in trials, and technology-based care strategies. You'll also hear about how you, as someone with FTD in their life, can join the FTD Disorders Registry to connect to opportunities to participate in research. First, while we know that awareness is growing, it still takes far too long to get an accurate diagnosis of FTD. I'd like to ask all of you who are diagnosed with FTD or personally connected to someone who has or has lived with FTD to answer a polling question about how long it took you to get an accurate diagnosis. While those answers are coming in, I just want to comment that we know that earlier diagnosis can give families vital information to help them make the most of precious time. AFTD and our partners, like the Diagnostics Accelerator and Target ALS, are investing heavily in biomarker development. Looking broadly, it's really an exciting time where the field of neurology has seen great advances in biomarkers over the past 5 years. We are hopeful that FTD biomarkers will be here soon to provide a vital stepping stone, both to faster, more accurate diagnosis, as well as to faster, better trials to develop the effective therapies that everyone needs. At this point, I am pleased to introduce one of our featured speakers. Dr. Adam Staffaroni is a neuropsychologist, assistant professor at the University of California, San Francisco's Memory and Aging Center, and a co-investigator with the North American ALLFTD study. His lab focuses on improving endpoints in clinical trials in neurodegenerative diseases, particularly FTD, and he has launched 2 studies on remote digital data collection using smartphones and sensors. He is here to provide an update on current major FTD research initiatives. If at any point you have a question about the topics Dr. Staffaroni will be discussing, please submit your question via the Q&A tab, and your questions will be answered where possible at the end of the session. Dr. Staffaroni, thank you for joining us.
Thanks, Penny. I'd like to start off by thanking AFTD for organizing this conference, for inviting me to speak, and for supporting a lot of the work that I'll be showing today. As Penny mentioned, I'm part of ALLFTD, a North American consortium studying FTD. And today I'm going to provide you an overview of that study, why we're conducting it, what are the questions that we're hoping to answer with this research, and ultimately how it will help patients and caregivers. I'll share some of our encouraging new findings and talk about some of our newest initiatives. These are my disclosures, nothing relevant to today's talk. So before I talk about the ALLFTD project, I want to give a brief overview of the importance of what we call natural history studies. So the natural history of a disease in medicine refers to the way a disease typically progresses without any intervention. So for FTD, these studies recruit participants and follow them over time. They typically collect information from a variety of sources, such as blood, images of the brain, and different types of tests. And then we analyze these different aspects of the disease to understand how they change over time. This helps us understand disease characteristics, improve diagnosis, and subtypes. It also allows us to identify the measures that change over time so that we can identify clinical trial endpoints, or measures that are administered several times during a trial to determine if a treatment is working. So for example, if a given test gets worse over time in some people, but not in others, it's not that helpful for understanding whether a treatment is working because some people would not have changed on that measure anyways. Natural history studies also have the benefit of developing a cohort of people that might be interested in treatment trials as they become available. So in summary, this work is setting the groundwork for success in therapeutic trials, both those that are here and those that are coming. So recognizing the importance of natural history studies for improving diagnosis and discovering new therapies, 2 projects were established in 2014. ARTFUL, led by Drs. Adam Boxer and Howie Rosen at UCSF, studied symptomatic FTD, both sporadic and familial cases. Lefties, led by Dr. Howie Rosen and Dr. Grabove of the Mayo Clinic, was a longitudinal study that followed families with familial forms of FTD, both participants with mutations and those who were family members but did not carry the mutation. And Artful and Lefties were successful by many metrics. So they established a network of 18 academic sites that enrolled 1,500 participants with longitudinal follow-ups that came to over 2,000 visits. And a lot of this data was contributed to national databases. A lot of the science that came from these studies was disseminated. And this included over 80 presentations at national and international meetings, more than 60 published manuscripts, and over 12 invited presentations. This is the cover photo for a special edition of Alzheimer's and Dementia, a widely read journal that's published by the Alzheimer's Association. And this special edition featured research studies that stemmed from the Artful and Lefties projects. I'll present a couple of these findings, which were very exciting, but at the same time confirm that we had more work to do. So this is an example of one of the studies from that special edition that looked at the familial form of FTD. We included 93 mutation carriers who had no symptoms or very mild symptoms, and we compared them to 78 participants who did not have a mutation but were from families with known mutations. We think of them as a control group who otherwise should have very similar characteristics to those who carry the mutation. The goal was to evaluate a battery of computerized cognitive tests that were developed with the purpose of being an endpoint for clinical trials. So in this graph on the right, mutation carriers are shown in red and controls with this dotted blue line. So you can see at baseline that this measure is able to detect some subtle differences, and that those carrying the mutation tended to perform worse on this over time. We also found that someone who declined on this battery tended to also show more severe disease progression and brain volume loss over time, which supported the notion that changes on these tests had clinical importance. However, we then went on to look at, if we use this test as the primary outcome for a clinical trial, how many people would we need to enroll to detect an effect? And these numbers are particularly high, particularly for a rare disease like FTD. So we concluded that we still need better endpoints, particularly for people with minimum symptoms. And we've made some adjustments to these tests that we think will give us better data in the new ALLFTD project. We also concluded that these estimates might be more feasible if we knew which participants were closest to symptom onset. So to that end, this is another study that we published in which we again looked at those who were asymptomatic or mildly symptomatic. For each individual, we calculated a score that told us how much their brains had shrunk compared to people of their same age. So in this graph on the right, we looked at whether this score told us anything about how likely someone was to convert to dementia over the years after their brain MRI was acquired. So in blue are people with low scores or not a lot of brain shrinkage, and in red are those with high scores. So this drop off here shows that roughly 50% of those with a high score converted to dementia within about 2 years. In contrast, for those with low scores, even 6 years later, only about 30% of people had converted. So this seems like it could be a really useful method, but we need a lot more data to really ready this for widespread usage. And there's many other outstanding questions. From the perspective of patients and families, people would like to know, when will someone with familial forms of FTD develop symptoms? For both familial and sporadic forms, how fast will the disease progress? And can we explain why the same disease manifests differently from person to person? For example, in familial forms, why do two members of the same family who have the same mutation, one presents with a motor phenotype and the other with a language phenotype? To answer these questions, researchers want to know whether there are any blood or brain measures that will be helpful. And for clinical trials, what should we be measuring? To help answer these questions, the medical field is very interested in something called a biomarker. So this is something that we can measure, such as brain imaging, proteins in the blood or spinal fluid, or digital biomarkers. It helps to indicate a biological process, so it's something we measure and, for example, could tell us something about injury going on in the brain. It could be diagnostic, so blood sugar for diabetes. Biomarkers can also indicate responses to interventions. So cholesterol measurements can show responses to a drug like a statin. And lastly, it can predict disease course. So in oncology, tumor size helps predict, in some cases, how the disease may go. And with these questions in mind, the Artful and Lefty studies joined together to become ALLFTD. This is led by Doctors Beauvais, Boxer, and Rosen. The goal is to establish a North American network of academic centers and a community of FTD investigators to continue to understand the FTD's natural history, develop biomarkers for diagnosis and prediction, identify new genes and mutations associated with FTD, and create an FTD clinical trial network. So this map here shows the sites that are participating in ALLFTD. We're now up to 23 sites around the country, including a couple of sites in Canada. And ALLFTD is divided into participants who will only have a single visit here in blue, and those with annual visits in purple. For people for whom it may be too difficult to come back year after year, they can do this abbreviated one-time visit called the biofluid focused arm. Everyone else, both sporadic and familial FTD, will enter into the longitudinal arm. Again, we're also including non-carrier family members, which allows us to distinguish between the changes happening that are due to FTD progression versus natural aging. So the hope for the longitudinal arm is to enroll 1,100 participants. We estimate that about 500 will roll over from the Artful and Lefty studies, and then we anticipate about 150 new participants per year. We'd love to enroll even more than that, but we do have some limitations on our resources and our staff. For this biofluid focused arm, the goal is about 1,000 participants. That would be about 200 per year, primarily symptomatic cases. So if you're interested in participation, what does it look like? So for the longitudinal arm, there's annual visits that last between 2 to 4 days a year, in which a participant and their study partner would come to one of these centers. For the biofluid focused arm, it would be a single research visit. Both of these arms would undergo a blood draw, which allows us to analyze DNA and RNA for genetic markers of the disease and measure blood proteins that may be biomarkers. The neurologic exam allows us to track changes in neurological features. And additionally, the longitudinal arm also collects MRI to get an image of your brain, some memory and thinking questionnaires, the ALLFTD mobile app, which I'll discuss shortly, and an optional lumbar puncture, which would allow us to measure proteins in the cerebrospinal fluid, again, possibly helping us find biomarkers. And I want to point out that we have a website in which you can find more information about participation in this study, as well as other resources. So this graph plots the years since the study began here at the bottom against the number of participants enrolled. So IRB approval occurred here in green, and then shortly thereafter, COVID restrictions hit. So you can see that compared to our planned enrollment here in this dotted line, we were really quite a bit behind in trying to get participants into this study, although we're starting to make up a little bit of ground here. And a very similar story for the biofluid focused arm, in which we're still quite a bit behind our enrollment targets. So to summarize the impact of COVID-19, it slowed enrollment a bit. We had to move a lot to video-assisted research evaluations. We've also moved a lot of questionnaires over to an online format. But some of the in-person evaluations are picking back up. This is really dependent on different sites and state regulations. And we designed and launched the ALLFTD mobile app. So even before COVID-19 hit, we were developing methods for conducting research at home. We think that for a rare disease like FTD, in which participants are spread out across geographic locations, that being able to conduct research remotely could be of great benefit. And remote solutions have the benefit of reducing travel burden. It allowed us to continue to collect data. And not only is it more convenient, but we think that it may actually provide us with additional data that can't be captured during a typical annual visit. So we think that by being able to administer these tests more frequently, we're going to get a more reliable estimate. We think that may help us be more sensitive to the earliest changes of the disease, and it allows us to collect some novel sorts of information like step count or how quickly somebody responds. So we've developed the ALLFTD mobile app, which is a smartphone application. And I think this is a really exciting opportunity for research participants to really inform the world of digital and remote data capture and assessment in FTD. And the implications are even larger, as this project will also help lay the foundation for digital assessments in a variety of neurological conditions. So the app is downloaded onto a participant's own phone. They complete questionnaires, cognitive tests, and then a motor and a language evaluations. Compared to the old-fashioned cognitive measures, it's much more gamified with up-to-date graphics, and participants receive Amazon gift cards directly through the app if they complete all of the measures. And this project has been supported by groups such as the Bluefield Project, the Tau Consortium, and the NIH. And we've developed the ability to collect speech samples through the phone by having participants complete a variety of tasks, such as describing pictures and reading passages. These tasks were developed in collaboration with an international team of FTD language experts and formatted for collaboration so other researchers can apply their state-of-the-art software to extract information about things like the pitch of the voice or the types of words that are being used. So here's an example of one of the tasks. Hopefully you can see this little video in which we have ducks, and the goal is to choose whether the center duck is pointing to the left or to the right. And we had participants take this test both in clinic and then again remotely at home. This graph here shows each person's score on their in-clinic and at-home performance to see how similar those scores were. Perfect similarity would be if every dot fell on this blue line, but this is about as good as we see in research, which really supports our confidence that these measures are reliable. On the left here, we show reaction time on that test with higher reaction times, meaning slower performance. Here in salmon, we have younger asymptomatic participants who performed the quickest. We have another group of older adults who performed a little bit slower, and then the symptomatic group that performed the slowest. So we see this stepwise increase that again gives us more confidence in the measures that we're collecting. And on the right, we see a nice correlation with age, such that the older participants are performing slower on this particular test. So we're now asking all FTD participants to take these tests, and I hope to share some encouraging results in the future. So I want to pivot and talk about FPI, or the FTD Prevention Initiative. This is a global initiative led by Adam Boxer at UCSF and Jonathan Rohrer, who leads the European analog to ALLFTD, which is called GENFI. This is an older map, but FPI includes Australian sites. We've added RedLat from South America and FTD Gens based out of New Zealand. And the goal is to promote responsible data sharing within the context of observational studies and trials in familial FTD, to create an international database of familial FTD research participants who might be eligible for clinical trials, and to create uniform standards for the conduct of clinical trials in familial FTD syndromes, and eventually organize global platform trials. So I want to give an example of some of the work that's really hot off the presses. This project takes advantage of the international cohort to answer some of the important questions that I outlined earlier. And we're really learning a lot from this, and it's clear that this project is going to benefit future trials. This project was made possible by the support of AFTD. It was conducted with the help of the Barry Statistical Consulting Group, and I have the other members of our team here who contributed to this research. So the goal of the study was to better understand disease progression in familial forms of FTLD. As I mentioned earlier, understanding progression helps us determine how big a trial needs to be, what outcomes should be used for these trials, and when will presymptomatic mutation carriers begin to show symptoms. So this study included data from an international sample of familial FTLD participants. So we included 1,000 participants, including almost 700 who were mutation carriers. And this focused on the 3 primary genes, the C9orf72 repeat expansion, progranulin, and mutations in tau. So on the left are the measures that went into this study. The CDR is basically a measure of somebody's symptoms based on an interview with their study partner. We include a lot of thinking and memory tests that were administered in both ALLFTD and GENFI. We have levels of a biomarker called neurofilament light chain that's measured in the blood. So on the right, you can see a neuron in the brain. The neurofilament is basically part of the backbone that makes up the neuron. And we find that in a variety of different neurodegenerative diseases, it actually gets released and we can measure it in the blood. And then we quantified the size of a few different regions of the brain. And basically we put all of this information into a statistical model, and through the magic of math, this model uses this information to determine how far somebody is from their symptoms beginning. So for example, if they showed no changes on any of the measure compared to the controls, the model would think that they were probably really far away from onset. And to try to make that a little bit more concrete, These are three separate subjects that went into this model. As you move from left to right here, it's telling you how many years the model thinks these people are from symptom onset. So -7 is 7 years before onset, 0 indicates onset, and then 4 would be 4 years after onset. So at the top here, we see this is how severe somebody's disease is based on their informant report. In purple is their blood measures of NFL, in green is the imaging, and orange is the neuropsych data. And so for this person on the left, the model thinks this person is about 5 years from symptom onset. You can see that all their scores are really pretty low and they don't have any clinical symptoms. Here's a person that the model thinks is getting closer to onset. You can see there's this elevation in their blood NFL and maybe a little bit of clinical change. And then somebody who's after onset has clinical change that's accompanied by changes in all of these different markers. And this figure is one of the results that we were able to pull from this type of modeling. As you move from left to right, again, it's how far the model thinks people are from onset with the different measures in different colors. And although a work in progress, these early results suggest that we're seeing these blood measures change many years before symptoms start. We're also seeing imaging changes here in green that also are occurring before symptoms as well. And again, this supports our enthusiasm about blood and imaging biomarkers. For example, NFL could be used to monitor treatment responses in the earliest stages where there's otherwise no clinical symptoms. And it may provide us with information about when somebody is going to develop symptoms. And NFL is already being used to track treatment effects in clinical trials for other neurological conditions. And those results were really consistent with another recent project that came out of this FPI initiative led by Julio Rojas, in which they looked at this same biomarker, NFL. They looked at people who were in familial FTD mutation carriers who were either asymptomatic or maybe had mild symptoms, and found that those who had high levels of NFL at baseline in red tended to show more quick worsening of the disease compared to those in blue who had lower levels. And then this was validated in the European GenFI participants. And really, there are many other studies that have also supported our interest in better understanding how NFL can be used. And to this end, the Bluefield Project organized the Neurofilament Surveillance Project, or NSP, which is an ancillary study to ALLFTD. This is also supported by the collaboration of multiple pharmaceutical companies. So basically, participants would enroll at their ALLFTD study site, and then a research nurse would actually come and visit them at the place of their choosing, such as their home, to draw a blood sample. There would be 13 visits over the course of 3 years, and these blood samples would be analyzed for things like NFL and other potential biomarkers. So you could see the study design here at the bottom, in which participants would come in for their annual ALLFTD study visits every year, but then we would be getting frequent measures of NFL from the blood throughout the course of the study. So similar to the app, we hope that more frequent assessments give us a finer grained understanding of really how these measures change and when they start to change. So recruitment for this study is underway. The goal for 2021 is to enroll 330 participants, and we're nearing about 100 at this time. So if this is something that you're interested in, you can talk with your site contact to discuss enrollment. So I want to thank GenFI for allowing us to include their data in these projects. I really want to thank all of the patients and the caregivers. It's really your time and effort that's made all of this work possible. I want to thank the funding sources, including AFTD. We also want to thank the FTD Disorders Registry. ALLFTD has partnered with them for recruitment outreach, newsletters, and more. And we'll hear more from Diana Wheaton about the registry in the next session. So thank you very much.
Thank you for spending time with us today, Dr. Staffaroni, and for such an informative presentation. We have some questions from our audience, and we have just a few minutes. So some of the questions were about eligibility into ALLFTD. So if someone's not an English speaker, or if somebody's not living near a clinical site, what would you suggest to them about their eligibility here and how they should think about participating in research in general?
Yeah, I think, we really have an eye towards inclusion and trying to offer our study to as many people as possible. I wasn't able to show the enrollment criteria during this talk, but, they're pretty broad. the primary languages currently that we're recruiting into this study are English and Spanish, although we hope to rapidly expand that out. But we are, we do consider the potential for enrolling people regardless of the language spoken into our study, often into the biofluid focused arm. And again, we're hoping to continue to expand that. And similarly, I think, for participants that are interested in participating from far away, I still would recommend reaching out to your study site. The FTD registry is a potential way to get involved. And we hope that the mobile phone application is going to help us overcome some of the limits. As I mentioned, this is a disease that is quite rare, and so participants are spread out everywhere. And so, being able to work with those regardless of their location is important to us.
Yeah, thanks. So for those who don't have a study site yet, they're not necessarily familiar with those, what would you recommend for them to first reach out? There is certainly the FTD Disorders Registry to help them find some of that initial information. What would you suggest to them?
Yeah, I think in addition to the FTD Disorders Registry, I would also suggest AFTD has a plethora of resources on their website. Actually, I think I was on the platform earlier and there were several resources listed and studies available to participate in right there on the website. And then I think I showed before ALLFTD.org to find out more information and find a contact point for this study as well.
Thank you. And the FTD Disorders Registry does have, I think, about 14 different research participation opportunities linked there. So we all do encourage you to take a look at that as well as at AFTD. So we also had a question about the intersection here with ALS. So we know that there are some genes that can cause AFTD that can also cause ALS. How is the natural history study, this ALLFTD network in FTD, coordinating with the longitudinal familial ALS studies about their work, say, on the C9 gene?
Yeah, so I think there's a lot of discussions going on between study leads of both of these studies. Certainly thinking about how participants with the C9 mutation can contribute to both of these studies. So I think there's a lot of overlap and interest in, sharing of ideas and the like. Yeah,
thanks. So, the last time we had a count of persons diagnosed with FTD was 10 years ago. So we have a question about, how accurate are our numbers on FTD? Do we know how many people are actually diagnosed? And if not, then are there any research studies going on where a new count of FTD is being included?
Yeah, that's a great question. So, as you're kind of alluding to, there's a lot of challenges with diagnosis. I've seen a lot of the concerns that were posted in the chat earlier today about being able to find a timely diagnosis. We often know that people are misdiagnosed first, sometimes as psychiatric disorders, for example. And so, there's been a lot of efforts to try to raise awareness, certainly over the last 10 years in the community, with other medical professionals as well. And so hopefully we're starting to get closer to more early and accurate diagnosis. But it still makes it quite challenging to conduct a real study of this. And my best guess is that the estimates that are currently out there are probably underestimates of how common this disease is. I think the more people are able to participate in this research, in these research projects, and I think that's another chance for us to continue to get more accurate estimates. I don't know of a exact study going on currently, but I'm sure there are people interested in this question. I agree that it's important to continue to revisit.
Thanks. Well, we just have 1 minute remaining. There's one or two more questions, but I'm not sure we can really cover them adequately. I think it's one of the key questions, and just to address on the point, one of the questions around, if you participate in a clinical trial, does that mean you'll be able to take the therapy after the trial concludes? And I think, I don't know if you want to just comment really briefly on that, because it's something that I think all the families are going to be wondering about, and is certainly so difficult and not something that the individual clinical sites often have any control over.
Yeah, I think there's a lot of thought that goes into how these trials are designed to make sure that the conclusions of this study that are made based on the findings are really solid, and therefore that if it is able to cause the kind of changes we're hoping for, that we can then feel very good about it and get it out into the community. I think the way trials are designed, it varies very much depending on the trial as to whether there's an open label. Sometimes there are extension phases that occur after the initial phases of a trial. So it's hard for me to answer that accurately. I would suggest if it's something that you want to know, reach out to the people that you're in contact with, or reach out to one of the sites that are recruiting and ask them about how these studies are designed and what the potential is there for continued. Okay.
Thank you, Dr. Staffaroni. And we are out of time, so we will now take a short break, and my colleague, Dr. Deborah Niehoff, AFTD Scientific Director, will resume the conference at 2:45 Eastern Time. Thanks so much.
Great. Thank you.